In Module 1, we explore the historical foundations of the field, the cellular organization of life, the differences between prokaryotic and eukaryotic cells, the role of organelles, and the nature of biomolecules. We also discussed how life's basic molecular units originated and are structured. In Module 2, we will deepen our study into the chemistry of amino acids and proteins, laboratory techniques that biochemists use to study molecules, the hierarchical structure of proteins and their biological applications, the chemical properties of water, the medium of life, concepts of acidity, alkalinity, pH, and buffers, and why they are critical for cellular processes. This module will equip you with both theoretical understanding and practical perspectives. 1. Chemistry of amino acids, proteins, and their derivatives. Proteins are arguably the most versatile biomolecules. They act as enzymes, structural elements, signaling molecules, transporters, and more. Their diversity arises from their building blocks, amino acids. 1.1 Amino acids. Each amino acid contains an amino group, a carboxyl group, a hydrogen atom, a side chain, a group, that defines its properties. There are 20 standard amino acids found in proteins. They can be classified based on the nature of their side chains. Non-polar, hydrophobic, e.g., leucine, valine. Polar uncharged, e.g., serine, threonine. Acidic, e.g., aspartic acid, glutamic acid. Basic, e.g., lysine, arginine. Amino acids link together through peptide bonds to form polypeptides. 1.2 Proteins and their derivatives. Proteins are polypeptides that fold into specific three-dimensional structures. Their derivatives can include glycoproteins, proteins with carbohydrate groups. Lipoproteins, proteins bound to lipids. Metalloproteins, proteins containing metal ions. The structure of a protein determines its function, and even small changes in sequence can dramatically affect activity. 2. Measuring techniques in biochemistry. To study biomolecules, biochemists rely on a variety of analytical methods. These allow us to separate, identify, and quantify molecules, and to determine their structure. 2.1 Cell fractionation. A technique used to isolate different cellular components by breaking open cells, homogenization, and separating parts by centrifugation. Differential centrifugation separates organelles based on size and density. Density gradient centrifugation refines separation further. 2.2 Chromatography. Chromatography separates molecules based on differences in size, charge, polarity, or binding affinity. Paper chromatography uses paper as the stationary phase, simple and inexpensive. Thin layer chromatography, TLC, uses a thin layer of silica gel on a plate, more sensitive than paper. Column chromatography uses a packed column through which a solvent carries the sample. Variants include gel filtration, size exclusion chromatography, ion exchange chromatography, affinity chromatography, high-performance liquid chromatography, HPLC, a highly precise method using high pressure to push solvents through columns. 2.3 Calorimetry. Measures the heat released or absorbed during biochemical reactions. Useful for studying enzyme kinetics and thermodynamics. 2.4 Spectrophotometry. Measures the absorption of light at specific wavelengths by molecules in solution. This is key for determining concentration and monitoring reactions over time. 3. Classification and hierarchical organization of proteins. Protein structure can be described at 4 levels. 3.1 Primary structure. The sequence of amino acids in a polypeptide chain. This sequence is determined by the genetic code. 3.2 Secondary structure. Local folding pattern stabilized by hydrogen bonds. Alpha helices, spiral structure stabilized by internal hydrogen bonds. Beta sheets, sheet-like structures formed by hydrogen bonding between backbone segments. 3.3 Tertiary structure. The overall three-dimensional shape of a polypeptide, stabilized by interactions between side chains, including hydrophobic interactions, ionic bonds, hydrogen bonds, and disulfide bridges. 3.4 Quaternary structure. The arrangement of multiple polypeptide subunits into a functional protein complex. Example, hemoglobin, composed of 4 subunits. 4. Determination and biochemical applications of protein structures. Protein structure can be determined using X-ray crystallography, provides high-resolution 3D structures. Nuclear magnetic resonance, NMR, spectroscopy, reveals structures in solution. Cryo-electron microscopy, cryo-EM, powerful for large complexes. Knowing protein structures allows scientists to design drugs that bind specifically to targets. Engineer enzymes with improved properties. Understand disease mechanisms caused by misfolded proteins. 5. Physical and chemical properties of water in biochemistry. Water is the solvent of life. Its unique properties are critical for biochemical processes. 5.1 Polarity and hydrogen bonding. Water's polarity allows it to dissolve many substances, making it an excellent solvent. Hydrogen bonds between water molecules give it high cohesion, surface tension, and heat capacity. 5.2 Role in biochemical reactions. Participates in hydrolysis and condensation reactions. Stabilizes structures through hydration shells. Acts as a temperature buffer due to high specific heat. 6. Acidity, alkalinity, and pH concepts. 6.1 pH and pH. pH measures hydrogen ion concentration, pH measures hydroxide ion concentration. Biochemical reactions are highly sensitive to pH, even slight changes can affect enzyme activity and molecular stability. 6.2 pKa and pKb. pKa represents the pH at which half of a weak acid molecules are dissociated. This value helps predict ionization states of molecules, influencing their solubility and reactivity. 6.3 Effect on cellular activities. Cellular compartments maintain specific pH ranges. For example, lysosomes are acidic to support degradation, while cytosol remains near neutral to support metabolic reactions. 7. Buffer solutions in biochemistry. Buffers resist changes in pH by neutralizing small amounts of added acid or base. Biological systems rely heavily on buffers to maintain homeostasis. Examples. Bicarbonate buffer system, maintains blood pH. Phosphate buffer, important in intracellular fluids. Protein buffers, amino acid side chains can accept or donate protons. Closing of Module 2. We've now covered the chemical and structural details of proteins. Core biochemical analytical techniques. The structural hierarchy of proteins and methods for determining them. The indispensable role of water. How pH and buffers maintain biochemical stability.